So thanks for the opportunity to just give you an impression of how we are looking at predicting the crystal structures of drug molecules such as these, where there's flexibility that can give them different shapes. But particularly, I'd like to give the impression of the interactions with experiments that are involved when you are doing something that is of interest, particularly to industry. This illustrates the problem. You have a transdermal patch used for treating Parkinson's disease, and you can see it started to crystallize out in a polymorph, a crystal structure of the drug that was not known and is more stable than any they knew of at the time they formulated the drug. This problem could have been solved, as shown by Tkachenko and a commercial company, by doing crystal structure prediction and predicting that there was this more stable structure to be found. So crystal structure prediction goes back to be based on the very simple theory that a molecule will crystallize in its most thermodynamically stable structure. This requires a search through all possible crystal structures, which is large. We can consider millions of structures with just one molecule and the rest of the molecules generated by symmetry into the unit cell. And the search space increases drastically with the number of independent molecules. This could be two different conformations of the molecule or the molecule and a water molecule, and with the degree of flexibility of the molecule. And we're seeking to find the most stable, which is approximated by the lattice energy, the energy to separate a static infinite lattice into the molecules at 0 Kelvin. But we would like to improve it to put in the free energy because the relative energies of polymorphs change with temperature. The initial idea of crystal structure prediction was to test this theory of predicting crystal structures, which even the existence of polymorphism shows isn't adequate, or to design new functional materials to see if the molecule will crystallize in a structure with the required physical properties, and then for pharmaceuticals and most of the specialty products, aid the polymorph screening and how they're developed because of this link between the structure and its properties, which may be functional, and also the spectra that you would use to identify which polymorphs you've got. And the processing, solubility is obviously a key point here, or the morphology of the crystals, and the overall performance of the product, of which the stability is a key one, and depending on your property, drugs, it'd be dissolution rate, etc. So to give an example of designing a new functional material, this was a great success in that Graham Day had got the trust of, sorry, I've gone back one. Graham Day got the trust of the experimental group such that they were willing to synthesize this molecule here for the first time on the belief it would crystallize in this highly porous material, which it did. And here is the output for the crystal structure prediction. Each spot is a minima in the lattice energy, and the one they were after was this one here, because the color coding is the dimensionality of the pores in the crystal, and the pores, when filled with solvent, stabilize it. And it was only a long time afterwards they actually got the most stable crystal form. The polymorphism is a huge challenge. This gives you an example of all the molecules, sorry, all the polymorphs that were known of this crystal structure at the time. And you can see that the properties are different, color and shape in particular, and there are a lot of differences. And there were some polymorphs, the ones down, I seem to have somehow got my computer in a twist. but it's not allowing me to use the pointer without changing the slide. Not quite sure why that's happened, but you've got a range, and later people were able to characterize two of these new polymorphs, which had two molecules in the asymmetric unit, and found another polymorph which had just one molecule and had been previously predicted. But you can see here that there are an awful lot of structures which have not been found. Yes, there are lots of polymorphs that have been found, but there are many more that haven't. And this is one of the embarrassments of crystal structure prediction. So here we've got a lot of polymorphs. They grew with time. This is another example of a polymorph screening, which was a long period done systematically with industry. It took them a long time, and we've eventually got the structures of 10 different polymorphs here shown on the crystal structure prediction diagram. This was difficult because this drug molecule readily crystallizes as a monohydrate, and they were really interested to see whether there was an unhydrated form that was competitive with this. In fact, it crystallizes with virtually every solvent and decomposes on heating. So only 2 of the polymorphs could actually be grown into single crystals to solve by X-ray diffraction. And a lot more were found by desolvating the solvates, and you had to solve it from the powder diffraction pattern. And we also used calculated solid state NMR for helping solve those structures. But the big embarrassment, you can see, is that red global minimum, which was not found and should have been found. And so they commissioned some work of crystallizing this molecule under pressure and did find another polymorph, but not the global minimum. So we wrote this survey of studies that had been published, done in conjunction with industrial pharmaceutical labs, and you get some sense from the names of using crystal structure prediction to help with their polymorph screening, characterizing forms, looking for disorder, as well as trying to confirm that they had the most stable structure and predicting possible polymorphs. But this over-prediction means that there's a huge question of which of these structures are artifacts of the crystal structure prediction method, particularly neglect of temperature, and I'm working with Matteo Servaglio on this, or are they undiscovered polymorphs, and can we suggest a non-routine experiment to find them? So the sort of calculations we're doing, some groups, including the commercial ones, now will do periodic density functional calculations on hundreds or thousands of low energy structures to get their relative energies. These methods have to be supplemented by damp dispersion, and they're only able to use fairly simple functionals such as PBE. And it's very expensive, and there's no hope of extending this to molecular dynamics methods. Our approach has involved doing electronic structure calculations on the molecule in every conformation. Firstly, to get the change of conformation and all the energy that it has, the cost it has paid to change its conformation, but also to be able to analyze the charge density to get the set of atomic multipoles to model the electrostatic component of the intermolecular energy. This is usually combined with an empirical 6x atom-atom repulsion dispersion potential that's been fitted to crystal structures, but I'm working with Alston and Mosquita on non-empirical methods for getting the intermolecular potentials. And the key is that these force field type calculations, they're not traditional force fields, have better prospects of being able to be used for molecular dynamics simulations of properties if we could only get an analytical form with intramolecular energy. Now, the differences between these different approaches to evaluating energies are sort of converging, but still there's a long way to go, as can be seen by this comparison of the 200 lowest energy structures produced by two searches. This is for this molecule ROI that I showed you, and it's comparing that search with one done by the GRACE structure. And you can see that there are only 6 of the polymorphs were found by both methods. One was a lot of structures aren't overlapping. And worst of all, the orange needles were outside the top 100 for our method and weren't even a minimum on the PBE dispersion corrected surface. Another embarrassment was our original search had the global minimum far lower in energy, and so were overestimating how much of a danger it was that that structure would be found. So it's important enough that there are blind tests of crystal structure prediction. They're planning the next one. And what I'd like to bring to your attention is that row attempted predictions of how many people tried to predict the crystal structure of each molecule versus the row below of how many of those groups had the correct structure in their 2 lists of 100 structures that they were allowed to submit. You can see it's a rather disappointing and challenging problem. The other thing that came out was from the drug company having given us 5 unpublished polymorphs, because a lot of people then went in to re-rank those energies. And you can see there an awful lot of different density functionals and dispersion corrections, making quite a difference to the relative energy ordering of those different structures. The dotted lines show the difference that you get when you put in an estimate of the free energy correction, and that also does a lot for the reordering of which of the polymorphs are most stable. So this is something where we're developing extra methods. We did some with Gerrit Brandenburg, and the leading method that's very recently been published, you can see how much rearrangement there is, but also now what they claim was the best method, the global minimum has not yet been found experimentally. So crystal structure prediction is depending on relative energies and relies on cancellation of errors. It's very dependent on the molecule, but also how it packs. This is strychnine, and you can see there's a big energy gap, no polymorphs, but we only get this case when the packing is uniquely defining all 3 dimensions. So it is relatively rare. Time's passing on, so I'll just skip over this way where we're using the predictions to design experiments to find predicted structures by subliming one molecule onto a crystal structure with the desired structure of the other. And also, we've been studying and funded for the effects of magnetic fields on which crystal structure form, based on having found the most stable form of coronine the first time in a magnetic field. And emphasize that a key issue here is the prediction of solubility. We can do this by a cycle of how much energy it costs to sublime into the gas, and then how much energy you get from hydration using liquid states. And this is very important for crystallization to separate out the 2 hands of a molecule. Some of you may remember the thalidomide tragedy, after which drug molecules have to be synthesized in one enantiomer form, and just give you that frightening graph of how little energy difference between the racemic and the enantiomer form there needs to be to switch over completely which one you actually see. And so there are a lot of small energy terms which we have shown can flip the balance of which structure crystallizes. So this is an area with so many challenges to theoretical chemists for developing methods. Even just at the level of our lattice energy CSP, we want to go to bigger, more complicated systems. This is an example of a drug molecule chloride salt that also has a dialcohol in it. Lots of different interactions to balance there. We want to be able to get our thermodynamics right. It's easy in the computer to apply pressure, not so easy in experiments. But unfortunately, our experimental colleagues cannot crystallize at 0 Kelvin, and we do have to put temperature in. And that's not easy. And overall, we really need to understand how these molecules crystallize so that when there is a polymorph that's predicted that you really want to get, you can develop a recipe for finding it for the first time. We've covered in the group a few hundred molecules now, and lots of fun stories. Crystallization is very molecule dependent, and it has been really, it's a continuing process of crystal structure prediction evolving alongside the experimental improvements in robotic methods of trying to seek new polymorphs and detect them and characterize them. And huge numbers of people have been working with me on this project because it is a balance. Experiments, computer algorithm developments, and line tests are very useful for getting industrial interest. And thank you for your attention.
Thank you very much, Sally. So Austin has a question for you. Would the seventh structure of ROY or ROY be found if PBE was used with a better dispersion correction, say MBD? So Austin, if you'd like to unmute.
I have unmuted. Can you hear me, Kevin?
Yeah, we can hear you.
Okay, good. So I mean, Sally, I mean, Karen has pretty much read out the question.
Yes.
What do you think?
No, in this case, it is frightening how much the relative stability of many organic crystals do depend on the exact dispersion correction. And you're right, MBD and MBD star are looking better in theory. But in this case, it was a delocalization problems in the PBE functional.
Right, so you have to go to PBE 0.
PBE 0, and that people are now beginning to, but it is very expensive to do periodic PBE 0 calculations. So those with a vast amount of computer time can do those on some key structures, but it is becoming realized that that is certainly an important step in some cases for that.
Thank you, Sally.
Thank you. Any questions from anybody else?
Yes, I have a question actually. Whether you guys have looked at some of these quasi crystals, or also there's another class of something between what may be liquid crystals and you know proper crystals, I guess, that we call, which is plastic crystals, where they have some disorder metrics in at least 1 degree of freedom. But what I mean is these are actually regular crystals with crystal diffraction patterns, but at least in one, let's say if they're long molecules, they may be able to have partial or full rotation along the long axis. Have you done some predictions on things like that?
Those sort of crystals are quite interesting in that is one aspect. That's one of the reasons why sometimes we appear to be over predicting, is that you've got a range of related crystal structures that actually, when you add temperature to them, would not be distinguishable because of these motions.
Right.
Yep. So it is often diagnostic of dynamic disorder, possibly dynamic disorder just in, say, the side chain or part of the molecule, that you can have different solutions that are pretty close in energy, that are related and may have very similar powder patterns, and therefore turn up in dynamic disorder.
Right. So we're doing a number of experiments on these type of phase transitions, and so I'd be very happy to talk a bit more about how we could combine, if you have some interest in them. Yeah.
Yeah. Yes. Particularly if if we've already done the CSP on that or related molecules, that could be a very quick.
Yeah. Have you done any simulations on alkanes, perhaps?
No, we haven't done much on simple alkanes.
But we can talk about other molecules as well. It's just those are probably the prototypical ones. Yeah.
Yes, because they go up into rotor structures.
Right.
But yes, it's quite a common problem that you have in the drugs, a small alkyl chain as a substituent.
Right.
Well, that in some polymorphs can rotate because there's room. In others, it's too well packed and doesn't. Yeah, thank you.
Thank you.
Karen, can I ask one more?
Go for it.
Just a short one. Sally, it looked like from, you were talking about the effects of free energy on these energy ranking of these structures. That one of the things which is missing from the kind of non-empirical or force field end is any finite temperature simulations of these crystal structures. What's stopping them from happening?
Well, I think that is still there. That's what we're working on. And there are quite a lot of people trying to do molecular dynamics. But the problem is having a good enough force field.
Okay.
To do the molecular dynamics. The molecular dynamics is very useful for cases such as the ones of the plastic crystals being referred to earlier, where you've got a lot of lattice energy minima sort of merge as you put in the temperature to be one crystal. And the big problem is the quality of the force field being good enough that such simulations are realistic enough in energy.
Mm-hmm. Okay, so this is a good place for the non-empirical force fields, right?
Yes.